Hello everybody, welcome back. Today we will be covering the electron transport chain. So remember in the last lecture when I was talking about the main metabolic path in the body, also known as the Krebs cycle, and one of its hugely important products that was created is NADH. And that is important because NADH comes into here, the electron transport chain. So the main function of the electron transport chain is to make ATP, but the other function is to reoxidize NADH and FADH2. And that will build the chemical gradient to have low pH in the intermembrane space, right here, and a high pH in the mitochondrial matrix. So we want more hydrogens in the intermembrane space. And this all works through the intermitochondrial membrane. And the general gist of it is that electrons will flow down the concentration gradient, creating energy that makes the gradient again and ATP. That's why it's called a chain. Right. So, the biosynthesis of ATP is using energy released in the electron transport chain, and the sources of hydrogen are NADH, succinate, and FADH2. So those sources are oxidized, and in the final couple reactions, oxygen is reduced to water. So before we go into the chain, let's learn the components pretty well. FMN is made from riboflavin. If you remember, that's vitamin B2, and that is a prosthetic group, remember, a permanent group of the enzyme NADH dehydrogenase, and that's a part of complex 1. We'll go through that later. Coenzyme Q is also known as ubiquinone, and that is an electron carrier. And what else to know? The outer mitochondrial membrane is permeable while the inner is selective, which makes sense. Free energy that isn't stored in the phosphate is vented as heat. So a lot of the cytochromes, or all of the cytochromes, they carry iron. And iron is important because it's an electron carrier. And this iron is different from our hemoglobin iron because this iron sulfate center in NADH dehydrogenase and they have a Fe3 plus, and that can be interconverted into Fe2 plus, which is kind of the opposite of our hemoglobin iron. But cytochrome A has a heme iron that has a free ligand, and that reacts with oxygen directly. And NADH dehydrogenase has the iron sulfate centers that transfers hydrogen to coenzyme Q. And here are the structures. Okay, so cytochromes. Cytochromes B, C, and A are our iron sulfate containing hemoproteins. And copper in cytochrome A is their final electron acceptor. Cytochrome, I just said in the last minute or so, but let's just reiterate. The cytochrome iron is reversibly converted from Fe3 plus to Fe2 plus, unlike the iron of hemoglobin. That is the opposite way. So iron plays multiple roles in the body, just depending on its form. So these are the complexes. Complex 1 is made out of NADH dehydrogenase, FMN, and the iron sulfate proteins. I skipped complex 2 because it's really only made of structural proteins. Complex 3 is made out of the ubiquinone cytochrome C oxidoreductase. Crazy long, I know. And the structural proteins, along with cytochrome B and the iron sulfate proteins. And then complex 4 is made out of cytochrome oxidase, the structural proteins, copper, and 2 cytochromes, cytochrome A and A3. Okay, so let's go through the steps. The first complex the general gist is that using NADH coenzyme Q oxidoreductase, it will oxidize NADH and transfer those electrons to coenzyme Q, which will be reduced. So remember the names of the complexes, because the enzyme name will tell you exactly what happens. So oxidoreductase. So you know this is going to be a redox reaction. The first component is going to be oxidized. The second will be reduced. This, in general, works for all enzymes, so useful tidbit of knowledge to file away. Okay, so that's the general step in the first complex, but it's a little bit more nuanced than that. So NADH is first oxidized, and then it will give its electrons to FMN. So the FMN is reduced into FMNH, which is oxidized, giving electrons to the iron sulfate, and then from there to the coenzyme Q. So it's a little bit complicated, but that's just the first step. And in doing all of those reactions, it is able to pump 4 hydrogens into the inner membrane space. And that is a energy required step. So once they pass all of these electrons to coenzyme Q, the second complex will it will oxidize succinate into fumarate using succinate coenzyme A oxidoreductase, but this can also be known in your textbooks as a succinate dehydrogenase. And those electrons from succinate will be carried to coenzyme Q. And then in complex 3, coenzyme QH2 cytochrome C oxidoreductase will oxidize coenzyme QH2 into cytochrome C, and it will just transport those electrons, and in that process it will pump out another 4 hydrogens. So right now we got 8 hydrogens floating around in the intramembrane space, so it is getting pretty acidic. So electrons are moved finally to the cytochrome A. That's the final acceptor on the complex 4. It's not drawn here, but it should be here. From there, cytochrome oxidase will oxidize cytochrome C and then form water. So it will pump out the final 2 hydrogens. So all in all, there will be 10 hydrogens pumped out into the intermembrane space. And then a hydrogen from the FADH2 to FAD reaction will remain along with the water. So not bad, right? So it's a 10-to-1 ratio. And from that makes the gradient. So the free energy changes occur because oxidative phosphorylation releases free energy as electrons move. That's important to the chemiosmotic hypothesis, where electron transport is coupled to the transport of hydrogen outside, making the gradient. As I went through all the paths, that was how the outside is more acidic versus the inside of the matrix. And when they go down through the concentration gradient, that free energy from the diffusion is used to make stored energy in the form of ATP. Right, so how do we do that? This is the final complex, complex 5, also known as ATP synthase, and that will make the ATP using the free energy of the proton gradient. So the pressure if you can think about it, is quite high right now. Of course, like the body wants homeostasis, it will want to diffuse in. And remember that the inner mitochondrial membrane is selectively permeable. So no matter what, these things can't enter. The only way it can enter is through this complex. So the hydrogen protons will re-enter the matrix by going through a channel, and then by that, it will cause the neck protein or the stalk to go through a conformational change, and then from that, it will rotate, and then the mechanism will change, and it will it will produce the energy needed for ADP to be phosphorylated in order to make ATP, sorry. And think of it kind of like a motor, right? So one comes in, turns, makes an ATP. Another comes in, another ATP. So it is a very efficient mechanism. And then with all those hydrogens coming in, it will start the chain all over again. NADH will be oxidized, and then coenzyme Q will be reduced, and then one thing happens after another, and then the chain will go. But it's not a free-for-all. There is regulations, of course. So let's just go back through the chain. Complex 1 can contribute to the gradient, but not complex 2 because of the components. So complex 1 has NADH and FMN, and that can participate in the redox reaction. I said before, but let's say it again, the H plus from FADH2 remains in the matrix. And 2.5 ATP is made per mole per 1.5 moles of oxygen consumed, and 1.5 ATP is made per mole of substrate. So these are actually pretty good. These are pretty good odds. This makes quite a lot of energy, as you can see. It is a very efficient mechanism. So all these reactions pump out hydrogen, like 4 hydrogens here, 4 hydrogens here, 2 hydrogens here, and it will build the gradient bit by bit by bit. Why, right? Like, why not just have like a big dump of hydrogen out and let all of it come rushing in like an action potential? Like, that would be efficient, right? That would make us create more energy quicker. Well, the body is quite intelligent, and because of the chemiosmotic hypothesis, that is why we don't make a big dump like that. It's because this is a lot of energy being produced, right? But the gradient is being built slowly, so that big dump of energy would not occur and the cell would not explode. So the electron transport chain itself is regulated by the ATP amount in the body. Well, of course, because if you have a lot of ATP, it's going to tell the mitochondria, hey, we have enough, don't make any more. And it's also regulated by hormones like epinephrine, which say, like, hey, we're in danger, we need to run, make some ATP, please. Another question that a lot of people ask is, why is FADH worth less? And the answer is in the electron transport chain. But first, FADH is just not a strong reducing agent compared to NADH. Why? It's just the way the structure is. It's just the way nature made it. And it's worth less because it binds to complex 2 and it just gives less energy. So the amount of protons is not like FADH2 doesn't make any protons that gets pumped out. So that is why it's worth less. Okay, so NADH can vary the amount of energy created in glycolysis or the electron transport chain or the Krebs cycle. You can see in the books it kind of deviates by like 1 or 2 ATP, not very much. And that's because of the shuttles used. So there are 2 shuttles that we should be aware of, and the first is the malate aspartate shuttle. That's where L-aspartate is going to be D aminated into oxaloacetate using some transferase, and then from there it's going to be reduced into malate, and NADH is going to be oxidized. So malate is important because malate can be transported directly into the inner mitochondrial space without sacrificing an ATP, because of the malate alpha ketoglutarate transporter. So it will accept the malate carrying all those electrons directly in, and then from there it will be oxidized, dumping its electrons, and then turned into oxaloacetate. From there, aspartate, and aspartate can be transported out using glutamate aspartate transporter that does not require any energy. This would just be like a facilitated diffusion out to go with its concentration gradient, of course. So this is efficient, as you can see. You don't lose any energy, and it's going to be in the organs that require a lot of energy, such as the kidneys, the livers, and the heart. And then the next shuttle is the glycerol phosphate shuttle, and that is where dihydroacetone phosphate is going to be turned into glyceraldehyde 3-phosphate, which can cross the membrane. So this is the less efficient shuttle, because when glyceraldehyde 3-phosphate crosses the membrane, it gets oxidized, and it loses an ATP when it's oxidized by FAD. I do not know why FAD is used instead of NADH in order to, transport the electrons. I'm just not sure why, but this is why this shuttle loses the ATP. And this is still an important shuttle because glycerol phosphate is non-reversible once FADH2 is formed. So once FAGH is formed, that goes into the complex 3, and then it forces the cell into the electron transport chain. So once it's made, the cell has no choice. Okay, so these are the diseases. They are not. Don't look up the diseases that target specifically the one part of the electron transport chain, but just know that the diseases are inhibitors of the electron transport chain or oxidative phosphorylation. One of the important ones are the uncouplers that will dissipate the electrochemical gradient. So the electron transport will take place, but not the oxidative phosphorylation. Defects will be caused by the mutations, of course, in mitochondrial DNA, and that will lead to the mitochondrial myopathies, which are diseases of the muscle. It usually involves like exercise fatigue, unnatural exercise fatigue, I should say. And another major disease is the Leber's optic neuropathy. Why is that? Is because the eye is, well, you should know, but like the eye is the most active muscle in the body. You know, you think it's the heart, but no, it's actually the eyes. The eyes require a lot of energy to blink, to focus on things, and to coordinate the movement. That's why we have two eyes, right, for binocular vision. But it does take a lot of energy to focus and to work concurrently. So if there is a lack of energy, it's not going to be able to work well, and that will lead to the neuropathy. And if you can see here, it's categorized by these like fuzzy looking shapes on the eye. And then a normal eye would look more like this kind of like orangey, pinky, yellow area. So we should know about the poisons. Poisons will inhibit the passage of electron by binding to the part of the chain, blocking the redox reaction. All the carriers before the block would be reduced, and after the blocks they will oxidize. So depending on which poison, it's pretty selective, and ultimately these will block ATP synthesis. That's the worrisome part. So, the drugs. Oligomycin will bind to the stalk of complex 5, and it will prevent the re-entry of H, or hydrogen. And of course, that's problematic, because how can the chain continue? Uncouplers will increase the permeability of the inner membrane, and that will reduce the electrochemical gradient. So a couple of examples are the 2,4-dinitrophenol that will make electron transport chain go without a gradient, and all the energy made will be released as heat instead of ATP. So this is important in patients that are going through shock. This is when we want them to, of course, increase the body heat. Like, this is important for people who are also going into hypothermia. So barbiturates will block the electron transfer in complex 1. And those would be like rotenone or teracidin or amytal and mercurialis and damarol. That will block complex 1. Antimycin and dermercurol will inhibit complex 3. And antimycin, you should know, is actually a antibiotic. Sulfuric acid, carbon monoxide, and cyanide will inhibit complex 4, and that will totally arrest respiration. Or, and by respiration, I mean chemical respiration, not like breathing, right? And then atractyloside will inhibit the transporter of ATP to ADP. So that will, there we go. It will prevent the creation of ATP and ADP because they will not be able to enter like the complex 5. Okay, I know that was a lot, but thank you for your attention and good luck studying.